Everyone. In this video, I'll talk about gene regulation in eukaryotes. So this is just an introductory lecture where I'll be discussing about the basic concept about gene regulation in eukaryotes. But before we talk about eukaryotes in particular, we should understand why a distinction has to be made between a prokaryotic regulation and eukaryotic regulation. For this, we'll again revisit the differences between a prokaryote and a eukaryote. And you know that basic difference between these two different types of organisms is the presence of nucleus in eukaryotes, where the DNA is compacted into the nucleus over here. On the other hand, in prokaryotes, DNA is in the open, it's in the cytoplasm. So this has an effect on the transcription and translation processes. In prokaryotes, the transcription and translation, they are going on simultaneously, or they are coupled together, as in lacoperon and tripoperon we have seen. On the other hand, in eukaryotes, because of this difference of nucleus, the transcription and the translation, they are delinked. Transcription takes place in the nucleus over here, and translation takes place in the cytoplasm. So because of this difference, there is an additional regulatory step introduced in the eukaryote. The other difference is the presence of in prokaryotes, the DNA is not associated with any other different kinds of proteins. On the other hand, in eukaryotes, the DNA is packaged into nucleosomes, and these nucleosomes are DNA plus these histone octamers, and because of this, they are organized into higher order structures or chromatin. Okay. Now this difference has an effect on the transcription. How it affects transcription? If suppose there is a DNA sequence over here, so this DNA sequence, let's say, is the point where there are certain proteins will come and bind to activate transcription. But because this region over here is associated with histones, the proteins cannot access these DNA sequences. So therefore, a way has to be found by which these DNA sequences are now free of histones, and this is done by two ways. Either the histones are modified, or the chromatin is remodeled. Both of them can take place at the same time also. Okay. Chromatin is remodeled. So this histone modification and remodeling of the chromatin has an effect on the organization of the histones or the chromatin. So now let's say, because there are different ways by which chromatin can be remodeled, which we'll not talk about in this lecture, but the final consequence of all kinds of chromatin remodeling is that this region over here, which was now associated with the histones, is now free. Now because this is free, the transcription factor can come and sit over here, and it can initiate transcription. Okay, so because of this difference, again there is the regulation of gene expression in prokaryotes and eukaryotes is different. Now another difference is the multicellularity of most of the eukaryotic organisms. Because of multicellularity, there is division of labor, and because of this division of labor, specific set of cells, tissues are assigned for specific functions. Now, for example, neurons, they have to carry out specific function. So, to carry out specific function, they have to express neuron-specific genes. Okay. On the other hand, muscles, they have to carry out a different function. So, in them, the muscle-specific genes are activated. Now these muscle-specific genes, they are not activated in the neurons, and the neuron-specific genes are not activated in the muscles. So this has again, this is done by gene regulation. Okay. The next is we talk about memory. What this memory is a way by which the daughter cells retain the information about which genes they have to express and which genes they do not have to express. It means always there is not a signal which will tell the information. Like, for example, if you have a bacterial cell growing in an ampicillin medium, so they will start expressing genes which are required for ampicillin resistance. However, when you remove ampicillin and if this divides into two daughter cells, these two daughter cells will not remember what genes were expressed in the mother. On the other hand, in eukaryotes, it's very important to have this information, because if, let's say, in a developing embryo, if this is the anterior side and if this is the posterior side, so cells which are present in the anterior side, they get information by various ways. There are morphogens, there are different factors which tell them that they are anterior side, and so therefore they have to express head or the anterior specific genes. Okay. Now these morphogens, they have a limited lifetime, so therefore they perish after some time. But still, this cell, when it divides into two and on further divisions, all these daughter cells, they will have the information about where they are present. So they will have the information that they have to express the anterior specific genes. So these morphogens are not required throughout the life. So this is important way, and this is an important regulatory step in the eukaryotes, right? Now there are the presence of the housekeeping genes and the luxury genes. Housekeeping genes are those genes which are required for all cell types for the basic functioning of a cell, and these genes, they do not need any additional regulatory step. So their transcription or their expression depends upon the turnover of the protein. As the protein gets turned over, so if the half-life of the protein is less, so genes are expressed continuously. So they do not have any other additional regulatory step. On the other hand, the tissue-specific genes, these are the genes which are expressed in particular tissue. So as we have seen in neurons, only the neuron-specific genes are activated, whereas in muscles, only the muscle-specific genes are activated. So these are regulated. Okay. Now the regulation of these tissue-specific genes, they are on a temporal and spatial manner. Temporal is on basis of time. That means there are certain set of genes which are required in the early development. So when they are required during development, they are expressed, then they stop expressing. Some genes which are required late during development. So depending upon the time during development or life of an organism, the gene regulation takes place. Spatial means in space. That means, let's say in kidney, in liver, in muscles, neurons, that's space. So they are regulated. Okay. Then there are the transcription elements. For whether it's a housekeeping gene or a tissue-specific gene, the basic transcriptional machinery remains the same. So if this is the transcription start site over here, so at around -30 base pair region over here, there is a Tata box. Okay, this is a consensus sequence which is conserved in all organisms, all genes, and at around -80 base pair there is a cat box. So these are few basic elements, promoter regions, which regulate the gene expression. It's at this Tata box that RNA polymerase will come and bind over here. Okay, RNA polymerase will come and sit. Now in eukaryotes, there are three kinds of RNA polymerases: Pol1, Pol2 and Pol3. So these Pol1 is responsible for the transcription of all the ribosomal genes except the 5S ribosomal gene. Pol2 is responsible for transcription of all the mRNAs, the messenger RNAs. It's responsible for transcription of all the microRNAs, some siRNAs, some small nuclear RNAs. Pol3 is responsible for synthesis of transfer RNAs, 5S RNAs, small nuclear RNAs, small nuclear RNAs. Okay, So distinct set of transcripts are synthesized by different polymerases. So when we talk about transcription, general transcription element, we'll refer to the transcription elements which govern the transcription by RNA polymerase 2. Now the RNA polymerase 2, it will not come and bind at -30 on its own. It needs additional set of factors which help in stabilizing the RNA polymerase. So the first set of factor which comes and binds to this region over here is TF2D. So TF2D, TF stands for transcription factor, 2 stands for RNA polymerase, and D is the name of that factor. Once this TF2D comes and binds, then you have TF2A and B coming and associating with over here. Okay. Then comes TF2F. TF2F also associates with Pol 2, sorry. It associates with Pol 2. Then you have, so once all these factors along with TF2E, H, J, so these are additional factors and they form a very large complex, and then once they are all assembled, the RNA polymerase will come and it will initiate transcription. So they constitute what's called as basal transcriptional machinery. They constitute the basal transcriptional machinery. Okay, so these are the basal transcription factors and they are necessary for the transcription of housekeeping gene as well as tissue specific genes. Now, what makes tissue specific genes different, expression different? Because apart from these basal transcription factors, there are other DNA elements over here to which different factors can come and bind and either increase the expression in a temporal and a spatial manner. Okay. For example, let's say this is a heat shock element. This is a heat shock element or HSE to which a heat shock factor comes and binds. Okay. So when the heat shock factor is bound with the heat shock element, then as the temperature increases, it leads to the activation of heat shock genes to produce heat shock proteins. Okay. To produce these heat shock proteins. So this is regulated, right? Now, as with heat shock elements, then there are certain steroid response elements to which the steroid receptors can come and bind and initiate the transcription. For example, glucocorticoid. So there's a glucocorticoid receptor which will bind to the glucocorticoid response elements and then initiate the transcription of the genes. Okay. Now, apart from these transcription factors and these activators, there's another set of activators and they are called as enhancers. Okay. As the name suggests, these enhancers, they enhance the transcription of the gene. But unlike all these other factors which are in close proximity of the transcription start point and they are always present upstream of the transcription start point, the enhancers function in an orientation independent manner and the location independent manner. What it means is these enhancer elements can be present on the upstream, they can be present on the downstream of the gene, and they can sometimes be present hundreds of kilobases away from the gene. So now the question remains is, if an enhancer element, if let's say an enhancer element is present over here, okay, enhancer element is present over here, so how does this element regulate the transcription of this gene from such far off when the transcription activator binds to it? Okay. Now the binding of the transcription activator to the enhancer element, it introduces a bend in the DNA. Okay, so this DNA is bent in a manner to bring this enhancer element close to this is the activator which is bound. Now this is close to the transcription start site. So these are the different factors that were bound, RNA polymerase. So there's interaction, and then the RNA polymerase can increase the transcription of this gene. Okay. Apart from these activators, there are other proteins which are called as architectural proteins. As the name suggests, they change the architecture of the DNA, they change the conformation of the DNA, so it helps in binding. Okay. Now, another question that arises is that if the enhancer element is situated so far off from the gene, so if suppose there is another gene in between over here, will this enhancer influence this gene? So the question is, will this enhancer influence this gene? And the answer is no. Why? Because the genes in between, they are bounded by certain elements which are called as boundary elements. Okay, they are called as boundary elements, or they are called as insulators, which insulate the effect of this enhancer on this gene. So therefore, the enhancers are very specific for the gene on which they are acting. Okay. Now, apart from these enhancers, there are repressors, and as the name suggests, they repress the expression of a gene. There are co-repressors, there are co-activators. So these are all additional set of factors which help in fine-tuning of the gene regulation. Okay. Now, what are the different levels of regulation? So the first level, of course, is the initiation of transcription. And as we had seen in prokaryotes, we have chromatin. So therefore, the initiation is a major regulatory step. So the chromatin has to be remodeled, the DNA element has to be free, only then the transcription can begin. So transcription initiation is the first step of regulation. Now once the transcription has initiated, it will elongate, it will terminate, and then it will form a nuclear pre-mRNA. We are talking about the mRNA genes over here. This nuclear pre-mRNA, and it has a specific intragenic sequences which are called as introns. So these introns are, they make these genes, what we call them as split genes. So you have exons which are interrupted by these introns. Introns do not code for a protein, so they are split genes. Okay. So the post-transcriptional modification, they include, first is capping at the 5' end by 5-methylguanosine, 5' end of the pre-mRNA. Then you have poly-A tail addition. Okay. The length of the poly-A tail can be between 20 to 200 adenine, and this is at the 3' end. And then there is splicing. The splicing, it means the introns are removed and final mature RNA that is produced has the exons. Okay. So this is the 5' cap and this is the poly-A tail at the 3'. So this is the final mRNA, mature mRNA, which does not have these introns. So these introns are spliced out. What you can look for is what is the importance or what is the advantage of a eukaryotic gene having introns, because they are ultimately spliced out. So this you can look for answers, and you will find it's very interesting what is the function of these introns. So now then the next is, once the mature mRNA is formed, it comes out into the cytoplasm. Once it comes out in the cytoplasm, then the next regulation can take place at the translation initiation, whether the mRNA has to translate or not. If it does not translate, the protein will not be formed, so that function is not going to take place. Then the post-translational modification. Now, an example of translation initiation, I can just tell you an example. In the oocytes of a developing of an egg, in oocytes of an let's say any organism, there are many mRNAs that are already produced. But these mRNAs, they are not translated because they are covered by certain proteins called as maskins. Only when the signal comes for the translation to initiate, these proteins are removed and translation begins. Then post-translational modification. This is a very important step where a protein which is modified in different manners, and the most common form of post-translation modification is phosphorylation. Okay, many proteins, they are activated only when they are phosphorylated. So phosphorylation step is again regulatory step. If a protein is not phosphorylated, even if it is present, it is not activated. Other kinds of modifications are sumylation, ADP ribosylation, ubiquitination, and so on. Then you have localization of transcripts and proteins. This is again an important step. What do you mean by this is that let's say this is a cell, and if an mRNA that is produced, it is present over here. And if this is a dorsal side and if this is a ventral side, so the protein which is produced by this mRNA, this protein is actually required in the dorsal side. So therefore this transcript, as long as it is present on the ventral side, it is not translating. But when the proteins are needed, this transcript goes to the dorsal side and then it makes the protein. And so this is how localization can regulate the gene expression. Another is localization of the proteins. What it means, as we have seen for heat shock factors, the heat shock factors, they are actually present in the cytoplasm. Only when the temperature increases, only when the temperature increases, these factors, they are activated. Different organisms activate these factors in different ways. But then once they are activated, they enter into the nucleus and then they start activating. So the localization of protein is again a regulatory step. Okay? So these are the different ways by which genes can be regulated. And one important regulatory mechanism in eukaryotes, we call it as combinatorial regulation. What it means is that a gene, apart from the regular promoter region over here, there are different elements to which different factors bind. A, B, C, D, different factors bind to different region. Now depending upon which set of transcription factors are bound, the gene activation will be temporally or spatially regulated. So if let's say A and C and D are bound, then this gene is expressed in tissue X at time T. If suppose B, A and C are bound, then this gene is expressed in tissue Y at time T1. Okay, so this is how the genes are regulated. As I told you, this is just an introduction. At each and every level, there are more fine tuning of regulation which helps a eukaryotic organism to carry out its function. So this is about gene regulation in eukaryotes.